Let's start chapter 17. Blood. What's blood?
It's a connective tissue.
Connective tissue, very good. Where's my mouse?
Which is fluid.
It's a fluid connective tissue, yeah. So the matrix of this connective tissue is what? Plasma. And the cells of this connective tissue are? Yes, yes. There's several different ones. There's red blood cells or erythrocytes. There's white blood cells or leukocytes. And that, of course, we can't make that simple. There's more than one leukocyte. How many different leukocytes are there? 5. And this is the confusing part of this wonderful chapter because the words are a little bit close. So leukocytes, all white blood cells. 5 different white blood cells. Neutrophils, lymphocytes, which sounds like leukocytes, but isn't. Different cell. Lymphocytes, monocytes, eosinophils, basophils. Don't write this all down. It's all right in your book. Calm your hand. Yes? And then, never let monkeys eat bananas. Exactly. I'll give you that little trick in a minute. And then, we also have fragments of cells. They're called platelets. Those are going to help with clotting, that we'll see in this chapter. So when I talk about whole blood connective tissue, it's got the same basic parts as connective tissue we learned about way back when. The matrix, in this case, is liquid. And what does blood do for a living? It's a transport system. Transport what? It transports nutrients. It transports respiratory gases, oxygen, and don't forget, carbon dioxide, also very important, and hormones that we just talked about, right? What else? Electrolytes, yes. Waste. So not only does it transport good things, it transports not so good things, yes? It's a important transport system. So that's why connective tissue, this is the truest of connective tissues, because it's connecting what? Everything together, okay? So when we digest things, it's taking those up and distributing them to all the cells in our body in some way, shape, or form. And then it's picking up all the waste products from metabolism and distributing those to where? Who gets rid of waste? Kidneys. Respiratory system. Skin. Okay, so these are some examples of where the waste is brought to, filtered out, and removed. So, physical characteristics. About how much do we have?
5 to 6 liters.
Yeah, 5 to 6 liters, depending on our size. The bigger we are, we're going to lean more towards the 6 liter. Yeah, size has a lot to do with that. Muscle mass has a lot to do with that. So between 5 and 6 liters of blood. The pH of blood is extremely important because not only am I distributing all of these materials and picking up waste, but I'm also the fluid that surrounds a lot of the cells where a lot of chemistry is happening. pH is very important. Brush off your brain and remember what pH is. What's pH?
It's actually the content of hydrogen.
Hydrogen ions in solution, yes? The definition of pH is the concentration of hydrogen ions in solution. So one of the huge roles of blood is to make sure that the distribution of hydrogen ions in solution, especially blood plasma, is distributed evenly throughout the body. So what's the optimal blood pH? Very tight, yes? 7.35 to 7.45, and that's extremely important. We're going to find out when we talk about plasma, not only does it have all of these other wonderful things, but one of the things we're going to talk about with plasma is buffers. And those are molecules that are either going to accept extra hydrogen ions if there's too many in solution, or they are going to donate hydrogen ions if there's not enough in solution. Those are the guys that are going to help keep the chemistry with respect to pH even. So,
Is this helping with the free radicals as well?
well, it's going to help distribute those molecules, those enzyme systems that we talked about. Those are going to be distributed in plasma as well. So, physical characteristics, volume, you should understand. If I took blood, whole blood, it's going to look red, right? But if I put it and let the heavy stuff, the cells settle, put it in a fancy little instrument called the centrifuge, which you will do in lab, you spin it down really fast, the heavy stuff comes to the bottom, and what you're going to see is 55% of that total volume is plasma. Not sure, that's up to Carmen. Sometimes I typically demonstrate, and then people who want to bleed can bleed in my lab, so it's up to him. You poke a hole in yourself. Finger stick. Finger stick.
You only need that much blood?
Yeah, yeah. You don't need a lot. No. Oh, see, she's one to sticking needles in you. Careful for her. So 55% is that liquid, plasma. Is the liquid water?
Yeah,
It's thicker than blood is thicker than water. That is true. Most of it is water. But the larger molecules that are being distributed, like the antibodies, that's one thing we forgot. Did we say that in our list? Antibodies, hormones, proteins, they make it physically thicker.
And protein.
Yeah, and proteins. Well, proteins, that's a general term because some hormones are protein in nature and some, all antibodies are protein in nature. So the proteins in there make it thick. The formed elements are 45%. Now, most of that 45% is red blood cells, erythrocytes. And when you take your little tubes of blood, they're going to be from a finger stick. They're in a little glass rod, and you spin them down. You can actually see a little tiny white stripe that sits on top of the red blood cells. It's called the Buffy Coat. And that contains what? Leukocytes and platelets. So white blood cells and platelets. So when we look at the cellular content of blood, what's most? Yeah, red blood cells. So, function. What's it for? Distribution. Doling things out. Transport.
This is actually the centrifuge.
Centrifuge.
This is how actually people make sour cream home.
Yeah.
Using centrifuge.
Yep.
This is how my grandmother always.
Yeah, because again, the heavy materials will flow to the bottom.
Like the hardest part will just be in the.
Yep, in the bottom.
In the bottom
And then get rid of the top.
Yeah. Actually, no.
Well, you can use the top for other things too.
Yeah, it can be useful for different things. Actually, can prepare. It's not very popular here in America, but very popular in Russia. Like how it's called buttermilk.
Oh yeah, yeah.
This is how they make buttermilk from
Right.
that portion. And it's yummy, but it's not really good here.
It's not well, and it's
Not
not
really
not
good
that
here.
tasty to drink.
But
It's
really
good.
much because they put it different, like
Yeah. Or cooking with it is good.
and, yeah, fruit or something.
Yeah.
It's good. It's like a yogurt.
I make good biscuits with buttermilk. It's good.
At one of my jobs, we use a centrifuge to spin urine samples.
Correct. And the same thing will happen in normal urine is liquid.
Right.
But you will pass some cells, maybe some heavier elements like crystals, and those will fall to the bottom. You look at a urine sediment when you do that. And you'll do that too, the end of the semester. You'll all have to pee in the cup.
Oh,
Oh yeah, we put you to work in these labs. Well, at least hopefully Carmen will put you to work. So transport all those goodies that we talked about. Also helps to regulate. So not only are we going to regulate things like pH, which we just talked about, but we're also, blood is very important in regulating water balance. Not only through the circulatory system, but in the compartments outside the circulatory system. So delivering those ions and water to the different systems that will help us with water regulation. And what comes to mind off the top of your head? Water regulation system?
You mean hormones?
No, water regulating system. Kidneys, the renal system. So they're going to work hand in hand to help maintain fluid balance. So within the circulatory system and outside of the circulatory system as well. And then protection. So we're going to protect the system by having a certain pressure within the circulatory system that's going to help with distribution. And then preventing infection by passing around wonderful antibodies. This is different because I forgot to change the version of the textbook. Sorry about that. But the one I'm going to refer to is the one in your book, 17.1 on page 633. So these are some of the things we're going to find in plasma. Plasma is mostly what? Water. 90% on average, plasma, water. But one of the other very important things that are going to be distributed are those electrolytes. Sodium, potassium, chlorine, bicarbonate, which is an important part of the buffer system, are also distributed through plasma. We call these the solutes of plasma. Remember solute, solvent? So these are the things dissolved in plasma. Plasma proteins like albumin, 60% of plasma proteins produced by the liver. And these guys are going to help with osmotic balance, water balance. Globulins, gamma globulins, these guys we're going to talk about when we talk about the immune system.
Do you have to memorize the proteins that are
Yep.
in plasma?
Mm-hmm.
All of them?
Yep.
Memorize what?
You don't have to memorize the specifics, but you should know that these are all in plasma.
Okay.
Like all
Just
of
no,
them that are,
not like how many percent?
No.
Or they do
No.
do you want us to know all of them that are on this table or the ones that are listed on
The one in your book,
Okay.
table 17.1. I think they're all the same there. I think they just break it up a little bit different. Another protein.
I was gonna say, I have one more question.
Yep.
Why the plasmas that are in Lisbon, why do they? This is just a question. Why do they pay you to take the water out of the plasma?
Why do they pay you?
Pay you to take the water out of the
I don't know. That's a good question for the plasma center at
Well, I'm just
Lewiston.
wondering what is so significant?
Well, look at all the. You tell me. Look at the list.
I know, but
Look at the list. So what can any of these things do for a patient if I take them out of your plasma and give them to a patient? And we're going to talk about this when we talk about this chapter. When you get blood in the hospital, you're not getting whole blood. What are you getting? Cells. Washed cells that I put back in the bag. You know, when you see that red bag hanging in a patient's room, that's just red blood cells. But there's tons of different things in plasma that a patient might need as well. I don't want to give a patient whole blood because that's too much volume in their circulatory system. So if I can take, and this is what happens when you donate blood. You go, you donate blood, it gets separated. So we're going to separate out the red blood cells. We're going to wash them with normal saline, and we're going to package them like that. Then we're going to take the plasma and possibly take some elements from that we could use for patients too. There's different clotting factors. There's different chemicals in your plasma that I can actually separate out and administer to patients. For example, patients with hemophilia. What's hemophilia?
They don't clot.
They can't, they're missing some of the clotting factors that we're going to talk in this chapter about. It's 13, 9, 13, I think it is. It's a combination of the two. But I don't want to give a patient all of this, but in order for me to get a decent amount of it, what do I have to do? Yeah, take out a decent volume of plasma from you. Now you can regenerate your own plasma fairly quickly. Most of it's water. Yeah. So if I can draw that volume and just draw the plasma and put back your red cells, and you can regenerate that every week, I can get what I need from my patients from your plasma. Does that make sense?
Yeah. So when you go to like Red Cross and donate blood, they don't separate it and put your
No, they actually prep it for separation.
But they take your blood and then they have their own like function. They separate the plasma out.
No,
They don't.
someplace else does.
Someplace else does. So eventually, whatever you donated gets separated to red blood cells and plasma.
Correct. Yep. And then, well, I'll talk about it when we talk about some other things, but there's a whole bunch of other things they do to prepare for donation.
For RAM, yeah.
So I'll tell you about them, though, as we go. So yeah, those factors, those chemicals, that's why donating plasma would be a good thing to do. It would be very helpful to a wide array of patients. The other thing we're going to find in plasma, besides those really important proteins, is nutrients, respiratory gases, hormones, and all of that good stuff. So understand what's in plasma and why it is important. Now, pay attention to this because you're going to be doing this in lab also. You're going to be doing a differential blood cell count, and you're going to be looking at a blood smear. Now, if I just took a drop of blood and put it on a slide, and this is exactly how I make a blood smear, you can see it in your lab book, and move it across the slide so it evenly distributes itself. At the very end part, I'm going to see a nice single layer of cells. Come on in. And that's what you're going to be looking at under the microscope. So when we talk about formed elements, this is what we're looking at on this slide. Most of what I see on this slide is what? What kind of cells? Red blood cells, or erythrocytes. The other there, but much less numerous cells are what?
White blood cells.
White blood cells. Now, there's different kinds. Remember the 5. And then way over in the corner, we see a little clump of them, and typically you don't see them in clumps. Typically they're kind of spread out all over the place. Those are the fragments of cells that I talked about. Those are platelets. Those guys are going to help with the clotting process. So that's what the formed elements part of blood is made up of. Yep, there's a bunch, and I'll explain all of the differences to you. But there's different characteristics to all of the different blood cells. One of the things I want you to pay attention to is these look different than that. How come?
Nucleus.
No nucleus. Red blood cells are, they're called anucleated. We sometimes describe them as biconcave discs because yep. Now why does it do that?
Outside and inside.
Correct. So why are they like that? When they were born, and where are blood cells born? In the bone marrow. The red blood cells had a nucleus. Before they get into the circulatory system, what do the red blood cells do to their nucleus? They get rid of it. So what are basically red blood cells? And that's where the nucleus used to be. Kind of collapses on itself in the middle. Yeah, but see, where the nucleus, envision this same cell that used to look like this. When I get rid of the nucleus, I look like this.
Isn't it easier for transport that way?
Yeah, it's just basically a bag of hemoglobin. Lasts about 120 days in circulation, and then we recycle them. The liver does a nice job of recycling for us. So, erythrocytes, red blood cells, are described as biconcave discs because of the fact that they spit out their nucleus. Contain hemoglobin molecules. And if I take a drop of blood, and here's another thing that's going to be confusing through this chapter. There's normal, what we call normal values for all blood components. And the numbers are changing all the time. So I go from one diagram to the next diagram, and the numbers change. So I'm going to tell you which one I want you to know. They're all sort of in the ballpark. You're going to see in your lab book different numbers. Are all of them wrong? No, they're all in the ballpark. It depends on how you measure these things. So, not to panic when it comes to specifics as far as numbers go. So, within the red blood cell, there is hemoglobin molecules. We talked about these molecules last semester when we talked about quaternary proteins. Is that ringing a bell? Big, huge proteins. What's a quaternary protein? Take a guess. Quarter. What's a quarter?
4.
Yeah. There's 4 and a dollar, right? There's 4 what here?
Heme group.
There's 4 heme groups, but there's 4 globular proteins that make up this very large protein. And then within each of these globular portions, heme, a globin, there's something called a heme portion. And that's what this little outpull is. Kind of looks like a frisbee in the diagram. But those little frisbees are the heme complex. What's smack dab in the middle of the heme molecule? Iron. This guy is going to help me carry respiratory gases. So for every hemoglobin molecule, how many respiratory gases can I carry? 4. Now notice I didn't say oxygen, I said respiratory gases. Because this molecule is going to help me carry not only oxygen, but it's also going to help me carry carbon dioxide. So when I breathe in, I'm taking air from the atmosphere, I'm bringing it down into my alveoli that are covered with capillaries, and gas exchange is going to take place. When I breathe out, the concentration of what respiratory gas is increased. When I breathe out, no, carbon dioxide. So when I breathe in, versus breathing out, we're going to see that there's different concentrations of oxygen and carbon dioxide. And hemoglobin is going to help me carry both. So if hemoglobin is carrying oxygen, what do we call it? Oxyhemoglobin, exactly. When hemoglobin gives up oxygen, what do we call it? Deoxyhemoglobin. And what do we call it when hemoglobin is carrying carbon dioxide? Carbaminohemoglobin. So this is the different forms I can find hemoglobin in. And it depends on what it's carrying at the time. So and here's another thing you're going to hate my guts for, but get over it. You're going to see these throughout the chapter. These are the maturation charts for the different formed elements. Excuse me. All formed elements come from one cell type. What's the name of that cell type? Yep, hemocytoblast. Or another name for it is the hematopoietic stem cell. And where do I find this cell type? Where's that immature cell? It's in the red bone marrow. So hematopoietic stem cells, hemocytoblasts, they're in the red bone marrow. And they have the ability to mature through some sort of stimulation. And can anybody, after the last chapter we just studied, take a guess at what might stimulate this cell to become a different cell? Some kind of hormone stimulation or some kind of chemical stimulation will help this cell mature into, in this case, a red blood cell. So,
do we need to know the
yes. Yes. You need to know the sequence. This is the easy one. Wait till we hit the white blood cells. You're really going to hate me. So enjoy the easier one right now. So, yes, you need to know the sequence. So as the cell matures, and what causes this cell to become a mature erythrocyte? Anybody know what the hormone is? Yep, EPO or erythropoietin. And who makes that? Kidneys. So that was in the last chapter, right? We're going to see it in this chapter again. And why might I need more red blood cells? Why would I need more red blood cells?
They last 120 days.
Well, they last 120 days, but they're constantly being stimulated to produce. Why might I step up production? Let's not be so dramatic. Oxygen, right? I'm actually going to step up when I am short of what? Oxygen in the circulatory system, when I'm short of breath somehow. So we don't have to put any holes in us to actually cause a stimulation. But when oxygen levels go down in the circulatory system, that's going to be the signal to the nervous system to have erythropoietin secretion from the kidneys. So, want to point out here, as we mature around the stage just before we go into the circulatory system, called the reticulocyte and the normoblast, can you see that at the end? What happens to the nucleus? That's when I spit out my nucleus. I'm still in the bone marrow when I do this. But that little reticulocyte is actually in circulation. So when I spit that cell out into the circulatory system, and how do I get it from the bone marrow into the circulatory system? No, it actually squeezes into capillary beds. When we talk about the venous system, we'll talk about different types of capillaries. And when we talk about the ones in the bone marrow, the capillaries have big spaces between the cells that create them. So the cells actually have to squeeze out of the circulatory system, out of the bone marrow and into the circulatory system. So reticulocytes is what comes on first, or comes into circulation first. And basically what's left in the reticulocytes are little bits of remnants of some of the genetic material that I spit out. Once I mature a little bit longer, I get rid of those extra little bits and I become the mature erythrocyte. What would an increase in reticulocyte count into circulatory system indicate? No, if I have, for some reason, I have too many reticulocytes, and there's a normal value for that as well, in circulation. What might that be an indicator of?
Stopping producing EPA, or EPOs.
No?
No, it stops actually, will not...
No. What you're... No. I'm trying to get at what would the bottom line be. If I'm pushing out too many reticulocytes, what do you think might be going on in the body? Or it could be I'm trying to make more red blood cells at a faster rate for some reason in the body. Either blood loss or maybe I have anemia, and we'll talk about anemia in a minute. But my system is trying to produce too many cells at too fast a rate. Sometimes if it gets really bad, we'll actually see nucleated red blood cells in circulation. And that's not normal. So we will see that sometimes.
Blood will be very viscous.
No, you'll just have the production rate and the push-out rate going much faster than it should. And it's usually blood loss that causes that.
Can I just ask you about phases development of erythrocytes?
Mm-hmm.
So here's like in I don't know in textbook of course, and in our PowerPoint there is a 3-stage. So do we need to memorize all this 3-stage without knowing the names for that?
You have to know the figure 17.5. That.
No, ours is a little different.
Yeah, that's what I was going to ask too.
Yeah, because it's
In your textbook.
In our textbook, it doesn't.
Yeah, this is the one in your textbook. Why is it different?
Because yours says early, late, and normal blast. Ours is
Oh, yeah.
vesicle.
Do the one in the book. This isn't up online anyway, so you don't have to worry about that. Yeah. Yeah.
Oh, yeah, we don't have normal blast on them.
No.
So
Right. You have orthochromatic erythroblast.
Yeah.
do we need to know like
Yes, from your book.
proerythroblast and
Yes.
erythroblast, erythroblast, and
Yes. Yes.
because in the book it's just saying just the 3 steps.
Yeah. In the
Like
book.
synthesis, hemoglobin accumulation, and injection of nucleus accumulation, reticulocyte.
Correct. You need to know the figure 17.5 in your textbook.
Is it for essay question or is it for essay question, a possible essay question?
No, it could be a regular question.
Oh, question, okay.
Multiple choice question. question. So yes, you have to know that. I told you weren't going to like me. All right. So regulation of production of red blood cells. Here we see one of our feedback mechanism. And it's dependent on oxygen levels in the circulatory system. So stimulation for increased production of red blood cells would be hypoxia. What's hypoxia? Yeah, low oxygen levels for whatever reason. Now there's tons of different reasons why there might be low oxygen levels in circulation. Anemia is one of them. Blood loss is one of them. There might be chemical reasons. You might be running in a marathon. All those reasons would fall into the stimulus category for low oxygen in blood. So we might also have a decreased red blood cell count depending on what's going on in our body, and we need to increase red blood cell count. So we're going to talk to the kidneys to release erythropoietin, and the circulatory system is going to come in contact with its target. And what's its target? Not bone. No, the hematopoietic stem cell. When you just say bone marrow, there's 2 kinds of bone marrow. You remember that, right? Don't just say bone marrow. Excuse me. So the hematopoietic stem cell, that's its target. It's going to cause that cell to then mature, go through this maturation process. And then more red blood cells are going to be spit out into the circulatory system and have the ability to carry that oxygen, and we are going to increase oxygen carrying capacity. So in your textbook they talk about dietary requirements, and basically these are the building blocks of hemoglobin. Iron, very important to make the heme molecule, correct? And when we talk about anemias near the end of the book, we'll see that that's a deficiency in either the shape of that hemoglobin molecule or the number of that hemoglobin molecule. So there's a whole bunch of different kinds of anemias that can be caused, and basically they all are a lower oxygen carrying capacity as a result. So you want to eat your spinach, but not just your spinach, because iron isn't the only thing. We also talk about B vitamins. And B vitamins are going to help with the chemical reactions that are going to help to build the hemoglobin molecule as well. So good blood food is iron and something that has B vitamins in it, especially B12. Where do I get B12 from? Does anybody know? Animals. Anybody vegan or vegetarian here? Which one? Which one?
I'm vegetarian.
You're a vegetarian. So what do you have?
I take vitamins. I take a B complex.
Do you eat eggs and milk?
No.
Okay. No.
Just vegetables. I eat a lot of mushrooms. I eat cheese.
You eat
I just
cheese.
don't like
Okay.
milk or eggs.
So you're good. You covered yourself with the cheese.
I eat a lot of yogurt too.
Yep. So you're good. That's tough when you create or you eat a vegan diet where you don't eat any animal products at all because of B12. So you have to make sure if you choose that lifestyle that you supplement. And the best is to go and get shots from your physician because oral supplements might not cut it for you as well. Unfortunately, where do they get the B12 shots from? Yeah, so just saying. Every once in a while. Yeah, do your research. So, what happens to that 120-day-old red blood cell? This diagram here on page 638 shows us basically the recycling of the components of red blood cells. What I want you to understand is that red blood cells aren't just thrown away when they're broken down. We recycle a lot of the parts. Liver plays a big role in recycling some of the parts. The other organ, spleen as well. Liver and spleen. They just talk about the liver here. But it's not the only player in recycle. So when you look at the cascade on, or the pinkish color cascade, you see hemoglobin is broken down into heme and globin. And the iron is stored as something called ferritin or hemosiderin. Some of the waste products of this breakdown is something called bilirubin. Anybody work in the hospital with any little infants, newborns? They get a little jaundice. What happens is when they're first born, they build up some of these waste products in the circulatory system because they're brand new to this whole thing. Sometimes clearing some of these waste products is a little bit of a problem. It takes a little bit longer than normal.
But why it gives the system like it's not normal, right?
No, it's normal. It's just their waste, their removal systems have to kick in. So some do and some don't.
But why most of like healthy child that they born, they don't have the yellowish color.
Right. It's because their removal systems have to kick in. It's not necessarily an abnormal thing.
It's not abnormal.
It's not, well, you don't want it the build up too high because it's toxic to the system, but it happens often.
You can see in the white of their eyes.
Yeah.
My son had to get a few tests after a couple days.
Right.
He had, like, jaundice.
So it's not life threatening, but it does have to be addressed.
If they got worse, the liver that's not functioning.
Or it might be over destruction of red blood cells. There's so many different reasons why, and it has.
The results she had to go under UV lights
Yep, that's what I was getting to, yeah. So the sunlight, UV rays, is going to help break down those products a little bit easier. So sunlight can help with this breakdown and removal process of some of these waste products. So
this is like similar to you explain it as like babies, but an adult,
Yep.
alcoholic.
Liver issues.
So is it increased with their bilirubin as well? Because I know like that's one of the things they focus on is the bilirubin in kids.
Exactly.
And I know that's regularly checked when you have like your annual urine test.
Yep,
They
it's
usually
part.
check the bilirubin as well.
Yep.
Just to make sure it's normal. So an alcoholic would have abnormal spiked numbers of bilirubin
Why?
because their liver's not.
Exactly. So
It's
decrease.
the same idea. They're not excreting the waste.
Well, they're producing more of it because of the liver damage that they're undergoing. So they're producing abnormally high amounts of bilirubin. Yes?
Are they producing them, or is it the fact that the liver just isn't absorbing them?
Right. So basically, the liver isn't able to
So
break
that's
down
the way it builds up.
exactly, and it builds up in the system. And you can see it it builds up in the plasma. If you take blood from a patient with cirrhosis or liver problems, the plasma doesn't look that straw whitish color. It looks more what? Yellow. And because blood's everywhere in the body, in the whiter areas like the sclera of the eye, you can actually see yellow as well. And that's because it's so high in the blood plasma. Does that make sense? Okay. So just a basic understanding of the liver and its role in breaking down these recycling products from the breakdown of red blood cells. We're also going to recycle some of the amino acids that make up those globular proteins as well. And then the large intestine is also going to help with preparing some of those products as well.
What do you want us to know from this figure? The entire system?
No. Just that the
we recycle the red blood cells?
right, and that the liver plays a role. Terms like bilirubin you should be familiar with. And the components of hemoglobin, heme and globin, iron. Now, the next part of the textbook talks about erythrocyte disorders. And there's a wide range of them. Anemia is a very broad term. And that means what? Yeah. It has something to do with decreased oxygen carrying capacity. That's what an anemia is. Now, there's a bunch of different reasons why an anemia might occur. So a basic definition of all the different anemias you should know. So blood loss anemia. If I get a hole in my circulatory system and I'm spilling out some of my 6 liters, I'm not going to have enough oxygen carrying capacity, am I? So that's hemorrhagic or blood loss. Not enough red blood cells produced. And again, for many different reasons this might occur. Problem with kidneys, for example. So patients on dialysis, patients with kidney failure in different stages of kidney failure might also be what? Anemic. Because the kidneys and their function isn't up to snuff. And one of the functions of the kidneys is to make what? EPO or erythropoietin. So that might be a secondary effect for patients who are having issues with kidneys. Renal anemia? Yes. The other one, iron deficiency anemia. Again, if I'm missing any of the building blocks it takes to make the hemoglobin molecule, it can result in less hemoglobin molecules. So they talk about iron deficiency anemia. Pernicious anemia, which is an autoimmune disease. Renal anemia, again, any kidney dysfunction that might result in the lack of the hormone that helps to stimulate erythrocyte production. And aplastic anemia, and that's mass destruction of what? Yeah, that bone marrow for whatever reason. Too many red blood cells. That shouldn't be an issue, right? That means we have amazing oxygen carrying capacity. Now it gums up the works, yes? Too many red blood cells, remember, have to move through things like capillary beds. And if you take a look at capillary beds, and I'm guessing that probably every single person in this room has seen blood moving through the capillary beds. Who hasn't? One by one, single file, red blood cells move through the capillary beds. That's how small the capillary beds are. So everybody moves through single file. That's how thin things are. If I have too many red blood cells in my circulatory system, my blood becomes too thick and things get caught up in those capillary beds. That can be a big problem. Some people try to do that on purpose. Have you ever heard of blood doping? Athletes try to do it to increase their oxygen carrying capacity so they might be able to endure for longer periods of time. It can result in fatal results, exactly, because it can cause spontaneous clotting to occur. So you don't want to have too many red blood cells. Thalassemia, Mediterranean origins, problems with the shape of the hemoglobin molecule. This is another problem with the shape of the hemoglobin molecule. This is called sickle cell anemia. This is, believe it or not, one mistake in the entire molecule. There's a problem with one amino acid at the 6th position. And it causes, yep. So it causes a problem with the shape of the molecule. When I describe the hemoglobin molecule, I describe it as a Ferris wheel. And respiratory gases jump on and respiratory gases jump off. If the shape of the molecule is wrong, the absorption of those gases can't happen efficiently in the respiratory system and at the tissue level. So the shape of the molecule is extremely important. Patients with sickle cell anemia end up having their entire red blood cells, many of them being sickle-shaped rather than nice and big and fat and round. And it's because of the shape of the hemoglobin molecules.
What does that do?
You decrease oxygen carrying capacity. So you tend to wear out, get tired faster, be tired a lot. Can't really overexert yourself too much because you have a decreased oxygen carrying capacity. Genetic engineering. But, now, and this is a strange little fact. See the shape of the cell? Normal cell round, sickle cells got that weird shape. There's a parasite very prevalent in Africa called malaria, Plasmodium falciparum. You'll learn that in micro class. And it hangs out inside red blood cells. But guess which red blood cells it doesn't like? Sickle cells. Guess where sickle cells thought to have originated? In areas that have a high incidence of malaria. So why would that be a beneficial mutation to someone? Well, it keeps them from dying from malaria, exactly. So they can live long enough to reproduce. This is what I'm trying to get through to my biology students. And they pass that mutation on. Not that it's necessarily a great mutation, but in the case of malaria and the infestations there, it allows patients to live longer to reproduce.
Is it always possible?
It's a genetic disorder, yes. Well, I don't know if it's a recessive or dominant. Is that what you're getting at?
Well, I'm just curious, I don't have it, but if I did, is it
I think it's a
high percentage that my child would have one?
yeah, I think it's a dominant trait. I think. Does it say?
I only know, that's
don't know. I'd have to Google that and let me know. The B12 thing?
Fortified cereal.
Yeah,
Deficiency, which pulls that out.
right. Yeah. So you have to supplement basically when you, yeah.
Liquid drops, multivitamins, B-complex capsules,
Right. But then it doesn't tell you where they come from. Right.
Yeah, I read that.
Yeah. They come from animals. But shh, don't tell the vegans that. Oh, is sickle cell anemia a dominant or recessive trait?
What do you want us to know about anemia?
Just a general definition of them all. So if I put not an essay question, but a multiple choice question, exactly, about iron deficiency anemia, that's pretty self-explanatory. You understand it's a low iron count. Polycythemia, too much what? Too many red blood cells, and they also mentioned blood doping there, which we talked about earlier. So those are the erythrocytes. There's millions of them per drop. Millions. So if I take a drop of blood and I put it in a little box, maybe a box the size of a cubic millimeter, and you'll see that's how they do the normal values in your book.
Sickle cell
Yeah, where is it?
is inherited from both parents. If you get the sickle cell gene from only one parent, you will have the sickle
Trait.
cell trait.
Yep.
You won't have the symptoms.
Yep. So it's a recessive trait. You have to inherit gene. You have to have 2 copies.
Yeah.
Okay. So what I'm referring to as far as cubic millimeters, a drop of blood, that's the table on page 644. Table 17.2. And this is, you don't have to memorize these numbers. I just want you to understand 644, 641, 644. Do you have, no. I'm talking about the normal value table. Like, wait a minute, they have the same book I do.
You said we don't need to know the numbers?
You don't need to know the numbers, but I want you to understand that there's a lot more red blood cells in a drop of blood than there is white blood cells. So when I look at the numbers of red blood cells per drop, it's in the millions. When I look at the numbers of white blood cells per drop, it's in the thousands or hundreds, depending on the type of cell. Yes?
But it's interesting how we can still see the leukocytes in the red forms.
Right. And when you go in and you do your lab
They're just taking
thing, your blood smear, you're going to see that you're not going to see what you saw on that thing. In each field, you're maybe going to see 1 white blood cell. You'll be lucky if you get 2, but you're going to have to search for your white blood cells. Yeah.
I don't think they will be like all on the
Yeah,
one
no,
side.
no. And that happens very infrequently when you actually look at a blood smear. Okay? So, lots more red blood cells than these guys. And this is the one that's on page 641. And these are the numbers you need to know.
You mean percentage?
Yep. So percentage of white blood cells, I want you to know from this chart. Because if you look at all the other ones, all the numbers are going to be different. So as far as normal values for the white blood cells, you need to know this.
So it's the percentages that we need to know.
Percentage of 100. And when you do your test in lab, you're going to do your white blood cell differential count. So basically you're going to go in, look at a blood smear, count 100 white blood cells. Now, the first one might be a neutrophil, and the second one might be a lymphocyte, and the third one might be a neutrophil, and another neutrophil. You see where I'm going with this? So you're going to count the white blood cells as you see them and differentiate them. When you get your 100 counted, these are the normals you should fall in. So of that 100, if I had 45 of them being lymphocytes, would that be normal? Yes. If I had 50 of them being neutrophils, would that be normal? Yes. Do you see that? And that's what you're going to do in lab. And that's what a white blood cell differential count is. So when we talk about the leukocytes, and again, be careful, because leukocyte, lymphocyte sound the same, yes? Leukocytes, all of them, all 5. So white blood cells fall into 2 categories. What kind? Yeah. Yep. Because the granulocytes tend to be phagocytic, and they tend to be more on the bacterial side, except for the eosinophils and basophils, which are parasitic. The lymphocytes tend to fight off viral infections. So, and we'll talk about that. Lymphocytes. So we have 2 groups of white blood cells, 5 altogether. Remember, never let monkeys eat bananas. That's going to solve 2 problems for you. That's going to tell you the 5 different types of white blood cells. And guess what else? The order in their number. So never the most neutrophils, let lymphocytes the second most. You see where I'm going with this?
Yeah.
Monkeys,
Well,
third
like
most.
the cranial nerves.
Exactly. So remember them, and that'll give you the order.
Mary money, but my brother say that business Mary money.
That's right.
I remember all
Yes.
of that actually.
So all those little things help you remember. Yeah.
It's easy to remember stuff.
So never let monkeys eat bananas gives us the 5 cells and the order in which they appear in number. Granulocytes versus agranulocytes has to do with what they look like under the microscope. Now, this one I have up here is bad. The one in your book is better. So look at the one in your book. The granulocytes consist of neutrophils, eosinophils, and basophils. And it has to do with what the cell looks like under the microscope. There's little bags of chemicals floating around in the granulocytes. And when I stain them, what does it make the cell look like? Yeah, granules or grainy. The neutrophils pick up a portion of a complex mix of stains called right stain. There's some neutral portions, there's some acidic portions, some basophilic portions. You see where we're going with this? So neutrophils have 2 very distinct characteristics. One, their granules pick up the neutrals portions of the stain, so they're kind of, I describe them as pinky purple, and they're much finer. The eosinophils pick up the eosin portion of the stain in their granules. This is a crappy one up there. It doesn't even look like an eosinophil, but the one in your book does. And you can see that the granules no longer look like sand in the background. They look like balls, big orange or reddish balls. That's the eosin portion of the stain. And the basophils, this is actually the one up here is better than the one over here. The granules are what color? Yeah, they're really, really dark. They're almost black. That's the basophilic portion of the right stain. So you see where they're getting their names from? Based on what they look like under the microscope.
You know what's helping me? I don't know, maybe some of you guys will help. The neutrophils, it's actually neutral because it has acidic and basic.
Right.
Then it's another, how's it called?
Eosinophils.
Eosinophils.
acid.
acidophilic base. So
Yes.
it's red. And then the last one, basophilic, it has a basic. And it's actually you can find it in the name. Base,
Right.
Base, basophil, so it's easy to remember.
Yep.
I think.
That's what I just said. Yeah. So, look at the granules. That's how you're going to tell the difference. It's really important that your right stain be balanced chemically, because if it's off, the colors are going to be off. And then you're going to have a problem distinguishing those cells.
So you said that the area that we're looking at on these are the darker areas?
Well, that's another thing I want to point out. Neutrophils, or granulocytes in general, have a very strange nucleus. What happens to the nucleus, which is normally round in many cells, it becomes lobar and weird shaped. You ready for the other name for, I'm going to throw it out there. Sometimes it's referred to as a polymorphonuclear leukocyte. Now wait a minute, big word. It's a big word, right? But break it down. Poly means what? Many. Morpho refers to morphology, shape. Nuclear, nucleus. And leukocyte, it's a white blood cell. So these guys have many different shaped nuclei. Polymorphonuclear leukocyte. So sometimes in the hospitals you'll hear them throw around, and this is usually some old school guy that'll throw it around, PMNs. That's what it means. Yep. Polymorphonuclear leukocyte. That's a neutrophil.
Know it all pretty much.
So you can just go, oh yeah, polymorphonuclear leukocyte, right? So that's a neutrophil. The reason we need the stains is because we wouldn't be able to tell the difference between the three at all unless we had the colors to help us, because they all look kind of the same. Exactly, and that's why you have to be careful for your stain. So when you work in a clinical lab in the future, you always have to make sure, A, you filter your stain, and B, it's fresh. Because it starts when the pH starts messing up, then you're going to start getting weird colors on your differentials, and then you're going to have a hard time
But it's not up to
differentiating.
us to provide a fresh.
If you work in the lab, it sure is. You've got to go change the stain in the machine.
Oh, there's a machine.
Oh, yeah.
Okay.
So, neutrophils, most numerous phagocytic cells. So if I have any nasty little particulates, like a bacteria, for example, I am going to see larger numbers of those cells. They're bacteria slayers, yes. Phagocytic. Eosinophils, we start to see large numbers of those for two reasons. And many of you, maybe in this classroom, have a little bit higher an eosinophil count right now. What might you be going through? Huh?
I hope they don't have parasites.
Nope.
In our body.
Allergies.
Allergies.
Anybody have seasonal allergies? You'll see higher numbers of eosinophils during that season. Also, parasitic infections will cause you to have higher numbers, but usually it's allergies. Well, no, but you'll pick it up, and if you do the differential count, you're like, oh, patient's got a little bit high. Sometimes when we start having problems with the white blood cell group on the whole, we'll start to see way, way high numbers, and those are usually some sort of leukemia. So that's really abnormal. Basophils. Again, those are the lowest in number. When you do your white blood cell differential, if you find a basophil, make sure you tell everybody in the class, because chances are you might not even find a basophil. They're so few in number typically. Again, we see these guys in their little chemicals have histamines.
So those are what's supposed to help my
Antihistamines is supposed to decrease the production of those guys. Yeah, supposedly. It just puts you all to sleep and doesn't really help. Another thing we have to remember is white blood cells serve a purpose in circulation, but most of them are just kind of hitching a ride in the circulatory system. Neutrophils can leave the circulatory system and go into tissues. When we talk about basophils, they can leave the circulatory system and go into tissues. We describe them as mast cells there. Excuse me. And this is where they'll release their histamine granules and cause an inflammatory response. The other type of white blood cells are agranulocytes. And why do you think I call them agranulocytes?
So they without granules.
Without granules. But don't get freaked out if you see some lymphocytes or monocytes with granules in them, because you will see granules in them. But not nearly as many as you'll see in the granulocytic cells. So the agranulocytes are the lymphocytes and the monocytes. Now lymphocytes, very different looking, yes? Big, huge nucleus. This is a small lymphocyte. Sometimes you'll see larger lymphocytes, but their nucleus tends to be nice and round, and also very, very dense. The monocyte is the largest of all the white blood cells. It also has a more uniform nucleus, but sometimes it can get a little carried away and kind of look like a kidney bean. It's not as densely packed. It tends to be more spread apart, and I describe it as airy looking, because you can actually see like little white dots in the nucleus. Its cytoplasm, the difference between the two is very different. The cytoplasm of a lymphocyte tends to pull out a lot of the very blue portions of the stain. And sometimes back in the day we described it as robin's egg blue. The cytoplasm of a monocyte tends to be on the grayish side, so not as pretty blue, and also not as uniform, so a little fluffy around the edges in the monocyte. So lymphocytes, what are they doing for me? Oh, you're going to love lymphocytes when we talk about the immune system.
Lymphocytes is
Yep, so there's two different types, and we're going to talk about them when we talk about immunity. There's the T cell, and there's the B cell. Yep, when we talk about viral infections and the need to produce neutralizing chemicals to help fight them. Anybody know what I'm talking about when I say neutralizing chemicals to fight viruses? Who? No. Normal. Neutralizing chemicals to fight viruses. Antibodies. Who's going to make antibodies?
B cells.
B cells. When you look at your maturation, and again, you're going to hate me for this, but yes, you have to know this. See this diagram on page 643? You need to know this, the whole thing. Yes?
Why do we need to know all the...
Because...
No, I understand. Like, you need to know that it will start from chemocytoblast, and then there's two different
Yep.
kind of like... But
Correct.
why do we need to know all the names for each cell? Why?
Why? Because I said so. How will that help you? It'll make you understand the maturation process of those cells.
But understanding and knowing the names.
Yeah, you can waste time fighting with me, but I'm still going to make you memorize them.
I don't want to fight. I just
Okay.
understand why do I need to know that. That's all.
Because it helps you understand those cells and their different origins much better. So, we have granulocytes, agranulocytes. When you look at this maturation chart, again, most of the names are the same. So you're really not memorizing a whole bunch, right? When you look at the bottom of the chart, you're going to see two sections. You're going to see what's in circulation. Not the very bottom stripe. The very bottom stripe is actually where? These are the guys that are hitching the ride I talked about. No, you're going to find those guys in tissue. Okay? So that stripe where it says A, B, C, D, F, A, B, C, D, E, F, where did the E go? Well, in your book.
Oh, in the book
Um,
it says A, B, C, D, E, F.
I know. So use the one in the book. I'm going to get rid of this.
So there's mono.
So you have, this is page 643, figure 17.11.
They're showing B lymphocytes and T lymphocytes.
Correct, and which they don't break it out here for you. So lymphocytes come in two types, B lymphocytes, T lymphocytes. When we discuss the immune response, we're going to see they have different jobs. The B lymphocyte can differentiate and go into tissues and become a plasma cell. Plasma cells make what? Antibodies. Okay? Again, when we talk the specifics of an immune response, you'll see how these two players work together.
What is effector T cells?
They are going to be in an immune response. We have helper T cells, CD8 and CD4 positive T cells that all are integrated in an immune response that help to stimulate B cell production.
So it's a
We'll talk about it. We'll definitely talk about it when we talk immune system. We don't want to throw it in there right now. But yeah,
What is it?
that's what I just told you what it was. Yep. So the other one that can also migrate out of the circulatory system is the monocyte. The monocyte is also phagocytic, very similar to the neutrophil. It can squeeze out of the circulatory system and go into tissues and become a macrophage. Exactly. So remember when you discussed connective tissue way back when, and you looked at the cells that belong in connective tissue, you saw a macrophage? That used to be a monocyte until it went into the tissues. The other thing, and I don't know if Carmen mentioned this, do you remember when you talked about osteoclasts?
yes.
They're the ones that break the bone matrix. Guess what they used to be? Monocytes. So they can also differentiate into osteoclasts when they're stimulated by hormones to do so. Okay? So 5 different white blood cells. We'll talk about the nuances between T cells and B cells when we talk immunity. Know that they're both lymphocytes. They're all agranular cells. And we will pick up with the production and lifespan of leukocytes when we meet again on Thursday.
I have a question. Are you going to make our quiz for this chapter due the day that we're having our test as well? Or are we going to...
What do you want to do?
I'd like to know which ones, like if you get any wrong.
So what if I have them do, how would I have it do, you want to do Monday?
'Cause I
Or
just want to
Sunday?
really go
Monday.
over it.
Monday.
On the weekend.
Okay. So I'll ask you again on Thursday. Maybe we can have it due on Monday, and that way you can look it over before
Can
Tuesday.
you possibly do the quiz for this chapter today, if possible, for us?
Yeah. I can open it today if you want, and I'll still have it due on Monday. Will you do me a favor and remind me?
No, I can.
Otherwise I'm going to forget. Okay, so who